Welcome to episode 119 of Real Health Radio. You can find the links talked about as part of this episode at the show notes, which is www.7, so the word all spelled out, SEVEN health.com/119.
Welcome to Real Health Radio. Health advice that's more than just about how you look. And here's your host, Chris Sandel.
Hey everyone, thanks for joining me for another episode of Real Health Radio. So this week's episode is somewhat of a hybrid episode. So when I do my solo shows, I tend to focus on just one area and take a deep dive into that topic. And then when I do my interview podcasts, I tend to bounce around with a guest and touch on their story and then touch on a number of topics that they're well versed in. Well, for this show, I'm covering just one topic, which is genetic testing. And over the last couple of years, I've seen this area get a lot more focus in the nutrition world. I first noticed it with people talking about the MTHFR gene mutation, but more recently, over the last couple of years, people have begun to talk about genetics and health in a lot more detail and starting to make food and supplement recommendations off the back of genetic results from companies like 23andMe. So it's a topic I wanted to cover and I want to cover on the show, and to do it in a balanced and a measured way, and to look at what's true, what do we really know, and what's just hype. The only problem is I'm not an expert on genetics. I'm not even just okay with genetics. I am a complete novice. And I read some books, I've listened to some podcasts on the topic, but I am a complete beginner when it comes to this stuff. So what I wanted to do was take the normal approach that I have with a solo show, but invite someone on who is qualified to talk about this stuff. And last year, Precision Nutrition released an ebook called Genetics, the Universe Within. And often the phrase ebook can have certain connotations, like it's something short or it's self-published because no one's going to publish it. But that is definitely not the case for this book. It's 400 pages. It's got 600 off references. It's a proper book that could easily be published, but PN just chose to give it away as part of their marketing. And one of the authors of the book was Krista Scott-Dixon. And Krista has appeared on the show before and is one of the most articulate and well-spoken people I've ever had the pleasure of chatting with. And genetics, it's not an easy topic to talk about at a novice level. And given my audience and the fact that I never talk about genetics myself, my assumption is that most listeners are going to be at this basic level. And there's so much science that you need to understand for it to properly make sense. So while this is a topic that I wanted to do a show on, it was hard to think of someone who would be able to do it justice in the way that I wanted the show to come out. So when I saw that PN released the book and that Krista was one of the co-authors, I reached out and asked if she'd be interested in coming on the show and chatting about it. And Krista responded that she would be, but only if we could include her two co-authors, because they were the ones who really knew this stuff in detail, whereas she had more of a role of putting it all together. And so that's what we ended up doing, a panel discussion on genetic testing. So the panel comprised of Krista Scott-Dixon, Helen Kollias, Dr. Helen Kollias, and Alaina Hardie, all from Precision Nutrition. And normally I cover people's bio during the introduction, but each of the panel does this during our conversation, so I'll skip over that bit for now. And as part of the show, we cover a lot of ground, and all with the goal of having this be useful and accessible to a layperson, even if this was the first bit of information they've ever heard on genetics and genetic testing. So we cover the basics of DNA and gene and the genome and what these are and why they're important. We look at other terms that come up once you start getting into the genetic testing world. So things like SNPs and polymorphisms and genotype and phenotype and epigenetics and so on. We cover risk and understanding absolute risk and relative risk and why these are so important when it relates to genetic testing. We talk about what we currently do and don't know. And currently, we know pretty little amounts. And despite the wild claims that are often made, at this stage, most of these claims are hype. And or they're about things that we expect could be happening in the future, but they aren't reality right now. And we then do talk about the future. So things like CRISPR-Cas9 and gene editing, and then some of the social and ethical implications of this stuff. And I'm really pleased with how this episode came out. When I was emailing Krista to organize it, I told her that I had some apprehensions and some fears about this, that sometimes with panel discussions, people just start talking over one another, or there's internet issues with connections dropping out. So I did go into this with a bit of skepticism, but it worked out much better than I expected. And this is definitely down to my guests and their ability to talk about a heavy and weighty, detailed topic in a very accessible way. I'd love to hear your feedback on the show. What were your thoughts? Is this a format you would like me to do in the future? Would you like me to do more panel shows on a specific topic? So please leave a comment by going to the show notes, which is www.7-health.com/119, or you can drop me an email, which is info@7-health.com. So without further ado, here is my conversation with Krista Scott-Dixon, Helen Kollias, and Alaina Hardie. So on the line today, I have Krista and Alaina and Helen. Hey guys, thank you for joining me on the show.
Hey, thanks for having us.
Thanks.
Hey.
So this is the first time for me doing a panel discussion for the podcast. And despite being an atheist, I'm praying to the internet gods to play ball and that we have a discussion without people dropping out and too much lag time. So today's podcast is going to be focusing on genetic testing. And it feels like this is coming up more and more over the last couple of years. And truthfully, it's something I'm fairly late to the party on and I'm still a complete newbie with. So I thought it would be good to invite some people on the show that know infinitely more than I do on this topic. And as a bit of a frame for today's episode, I have a handful of goals that I'm hoping to achieve. And one is that I want this to be genetic testing 101. So if you're completely new to this topic, I'm hoping that you end this episode understanding a lot more than when you first started. I want this to be useful for health conscious individuals who've heard of genetic testing and are wondering about whether they should do it themselves. Maybe there's some anxieties or questions you have, or maybe you've done some testing and you're a little unsure about the accuracy of the results or what they really mean for you. And then finally, I want it to be useful for health coaches who have maybe heard of genetic testing and are wondering whether they should spend the hours and the time delving into the topic to become knowledgeable, to see if it can then help their clients or their patients. So this is what I'm hoping to address as part of this show. And to get things started, let's have each of my guests introduce yourself and share a little about A, your background, and B, how you spend your day-to-day. So Krista, can I start with you?
Yeah, I'm actually the least qualified person to be on this panel, to be totally honest. Last January, John Berardi at Precision Nutrition contacted me and said, would you like to write a book on genetic testing? And I was like, well, my last course on genetics was in 1991 in first year university. And I think science might have changed a little bit since then. So my role here at Precision Nutrition and in this book was really to be a researcher and a writer and to help organize the ideas of experts into a kind of a coherent framework. So I'm like a translator and organizer and researcher and kind of a developer of ideas to kind of make the science shine is really my job. And then also to think about, from a coaching perspective, like, what do people do with any of the information they get? So in my day-to-day as a coach, I'm always translating research into practice, and not just general practice, but practice that like real actual people in everyday busy lives can use. So that really is my day-to-day is researching and then taking that research and trying to translate it into real life practical stuff that you can do at 5 p.m. on a Friday kind of stuff.
Okay. So Helen, do you want to introduce yourself?
So I guess I have the most, academic background in the area. I did my doctorate in molecular biology, and there was a lot of genetics along the way, directly and indirectly. And my day-to-day is basically translating a lot of scientific ideas and concepts and new science, I think, in how scientists see new research and new developments and what they really think of things versus what I think many people perceive was going on, or how, generally it's being presented. Like you have a study comes out, it's like broccoli's going to kill me, or broccoli's going to kill me because I have X genotype. And it's like, well, not so much. That's not really what they found. So that's kind of my take right now, is more of kind of from a scientist perspective, trying to translate the science, and then and what we're trying to do as scientists, and what we're trying to get to, and what it means for the average person. And usually it's like, you have to wait. It's basically what it means most of the time. And so, yeah, that's my...
Okay. And then Alaina?
Yeah, so I am actually self-taught in molecular biology and genetics. I went to Singularity University in 2011. And through that, I discovered that molecular biology is like orders of magnitude more interesting than I thought. Like there was this moment where someone was like, I kind of grokked the idea that like, wow, proteins are like little machines that do things. And DNA is computation. And so my background is computer science. And I was like, oh, wow, this is like crazy interesting. So I became obsessed. And, I was a teaching fellow for biotech and bioinformatics. I did a lot of the, stuff with Helen on actually doing genetic testing and explaining the backgrounds, like how everything works. And I just freaking love molecular biology. I mean, that really kind of sums it up. And it's like a crazy thing to say, but it's absolutely true. And my day job at PN now, I mean, like we don't really have like job titles, we have roles. And so most of the roles that I fill right now are related to cybersecurity. But I've done lots of different stuff at PN. I've been at PN almost as long as Krista has.
Okay. Wow. Okay. Well, thank you all for that as an introduction. And with the questions today, I've got the three of you sitting here, and I'm going to direct the questions to the person who I think is best to answer it, but I could easily be getting this wrong. So if I ask a question and you think someone else is better equipped, then just pass it on. Or if I don't direct the question at you, but you think you have something that you want to add that the other person hasn't, then also feel free to do so. So the basis for most of today's conversation comes from the book called Genetics, the Universe Within, which, Krista, you reference, which you all co-authored. And you touched on it, but like when writing the book, what was the goal that you were hoping to achieve with that? And Krista, do you want to answer this one?
Yeah, and I think you actually sort of got at it in your introduction, which is basically to say, what do we know about genetic testing? Like, what do we really know? Not what the promise of it is by genetic testing companies and this sort of thing, but what do we really know? What does the science say? How does this work? So first of all, how can we help get all of our readers, our clients, our fitness professionals, other healthcare providers who might not have a background in genetics, but who might be working with clients, interested people, whoever, on the same page with a basic understanding of genetics and genetic testing, and to help people understand, first of all, what it is and what it does, and then second of all, how it applies to them. Because I think that's where the gap is for a lot of people. A lot of people either want to know, am I special? Spoiler, you probably aren't. Or if you have some rare genetic disease, it's probably taken you out at the 4-celled life stage. Or people want to know, how am I going to die? Like, I think those are the two fundamental questions that people have. And really, unfortunately, genetic testing can answer neither of those for the most part. And so this leaves us with the problem of trying to take what we do know, which is kind of cool, kind of interesting, promising, speculative, and helping people understand, okay, human variation is a thing, but what are you supposed to do about it? And so the book really explores, first of all, this really cool new science and what we do actually know for sure, which is very, very little, what we could know in future, how we could apply it, but also some takeaways to say, okay, if this is you, then here's what you might do. And I'm going to just wreck the surprise here by saying that most of the advice regardless of your genetic makeup, is going to be the same. So maybe you have a slight amount of variance in how your body stores fat or uses energy or whatever, but by and large, the rules of biology kind of still apply to all of us. No one is like a silicon-based life form. No one is using methane for energy or anything like that. So it's to kind of demystify it for people, but also so to take the scary parts out, but also to bring in the joy. And you can probably hear it in Alaina's voice. There's so much cool stuff in genetics and genetic testing. And for me, one of the big pieces was human variation, right? And I know a lot of clients come in and they're like, what diet is best for me? And they're assuming that there's one diet, there's one workout plan, there's one way of living that's going to be the best. And what genetics shows us is that we're so incredibly diverse. There's so much variation. There's so much complexity that there's no such thing as best. There's no such thing as ideal for everyone. So in writing this book, that was all of the things that I was trying to communicate and put it in language that people can understand. So it's not technical jargon. It's not completely obscure. We do get a little technical and we try to explain it, but to communicate it in a way that's accessible, interesting, I think occasionally funny. I don't know. I put some good jokes in there, but really, really invites people into the subject area.
Okay. And how long did it take from start to finish?
Help me out, guys. Would you say it was like 6 months or 8 months?
Well, it got released in like August and we started in January, but we had been talking about this, like Helen and I had been talking about it for like months and months beforehand. But like, this is kind of like one of the truisms of precision nutrition is when you put Krista Scott-Dixon on a project, it gets done really fast. So anything that we were going to do was like accelerated because Krista was involved.
Okay. And because of the kind of area that is where I imagine there's lots of new research coming up, how much did you have to change or rewrite? Because, I don't know, some new research came out and invalidated something that we thought was correct, or something came out that then opened up some new area of research.
Maybe Helen can answer this as well, but for me, it was, I'll just like briefly say, there's not so much that if you look at single studies, then yes, it seems like a big mishmash of stuff. But for us, we were also looking at the bigger picture, like what are the broad trends and what are the broad principles that organize this? And one of the broad principles, and Helen can go on about this for like 4 hours, I'm sure, which is that biology is complicated. Would you agree, Helen?
Yeah, exactly. And so I think what happens, and I think that this is, where the lay person gets confused when, and because I think also the 24-hour news cycle where it's like we have to put out new information. So it's like new study came out that says that all the other stuff is wrong. And so I read it, I get, my mom calls me, is like, I saw this on the news, and I'm like, oh, Mom. And so what happens is if a new study comes out, I'm like, well, that's interesting. I want to see like 3 more studies supporting this finding. If it completely negates everything else, if it's a continuation, then it's like, okay, well, this makes sense. And, 10 other labs and 15 other studies have supported this idea. But if you're flying in the face of all this other stuff, I'm like, I think I'll wait and see. And I think that's, I guess, general advice with science in general, like wait and see. If something's been around for 1,000 years or 100 years, then you're like, I'm pretty confident that this is true. If it's been a week, I would hold off before I change my entire life.
Okay. And so look, let's then imagine that this is the first podcast or bit of information that someone is ever hearing about genes or genetics or genetic testing. Alaina, would you be able to give a bit of an overview about what this is all about?
I can give it a try, with the caveat that, like Helen and Krista have said, like it's an incredibly complicated thing. But at its basis, we've all heard of DNA, right? And DNA is like this really long organic molecule that is actually a code. Like if you read the DNA, there's information in that. It tells the body or cells how to make proteins, for example. And so every living organism, like anything that is considered living, like yeast, bacteria, humans, rose bushes, elephants, they all carry around this set of plans, like in almost every cell in the body. And And so that full set of plans, like the sum of this DNA in a cell is called the genome. So that's what the term like genome means. It's like the entire collection of your DNA in an organism. And so the genes themselves, when we talk about a gene, we're usually referring to like a section of the DNA that represents the code, like the instructions for creating a protein. But I mean, it's different with different organisms, but those genes in the space of a genome are usually like a tiny fraction of the genome. Like the protein coding regions of the human genome is like 1% of the genome. So there's a lot more in the genome than just the instructions to make proteins. Like there are sections that say, okay, you know, like the proteins that are making copies of the genome, like attach here. And there are things that will affect how genes are transcribed. Like there's just this huge, I mean, there's something like 5 to 10% of the human genome is retroviral elements. So retroviruses are kind of viruses that like will infect a cell and then insert their own genome into the host cell. And so some of those things are really useful. Like there was, I mean, I guess I don't have to go into what that is, but that's like more than the 1% of our genome that codes for proteins. Like 5 to 8% actually come from viruses that infected our ancestors like hundreds of thousands or millions of years ago. Like that's fascinating, right? And so what we do in genetic testing is that we use different technologies to read certain parts of the genome that we know are interesting, that we think are interesting. We've identified things that are interesting about them. And then we can do sequencing, like whole genome sequencing, where we can read the entire genome from beginning to end. And that gives us like a different view than like the little glimpses that we have at it. And genetic testing is useful for tons and tons of different things. Like you can use it for tracing ancestry. So, you know, if you've done 23andMe or Ancestry.com, you might find that you're, you know, 8% sub-Saharan African and 14% from the Indian subcontinent and, you know, 28% from Northern Europe. And so we can do stuff like that if we can read the genome. You can look for diseases, you can predict traits. You can even capture serial killers. Like there was a serial killer who was captured just last week, I think, in the US, and they used DNA, like these genetic tests to like, they tested some old DNA and they're like, oh, this guy uploaded his DNA to one of these shared sites and then they found him. So, and we at PN are interested in discovering, like using this testing to discover what it can tell us about who people are, how their bodies work, and how we can serve them better.
Okay. And so one of the terms or the acronym SNP that's pronounced as SNP, this comes up a lot when people are looking into genetic testing. So are you able to explain what this is, Alaina?
Yeah, for sure. So you hear this a lot in like commercial genetic testing, and SNP means single nucleotide polymorphism. So the genetic code is like the series of letters. I'm sure that most people are familiar with the A, C, T, G letters. And so a single nucleotide polymorphism is like, instead of an A that's usually present, it's a T. And so that can like make no change at all to what happens, or it can change dramatically what happens. And so we've taken a survey of like an organism, like a species, and we're like, okay, this one little mutation shows up in people of sub-Saharan African descent. And it might or might not be associated with anything. So some of them are associated with risks, like your likelihood to get breast cancer. Some are associated with traits like whether you hate cilantro or, you know, what consistency the earwax is in your ears. And we like these things, like we like SNPs because once we know about them, once we know that there's like a little letter in the genome that can be different between different people, they're pretty easy and cheap to find. So most of the commercial services test for this like range of SNPs, and like anywhere from, I don't know, 250,000 to 5 million of these little individual letters in the genome. And they can be kind of useful, like especially in research. And I also think it's important that like one of the things that I wouldn't say stresses me out, but kind of stresses me out about commercial genetic testing and the way that we talk about it, is that this is really easy to look at. So this is what we look at. And it's like a small fraction of the genome and a small fraction of the story of what the genome does.
Yeah, and just to piggyback on that is like, there's a huge bias to genetic testing that looks at SNPs. And it's not even that we're only looking at specific region, like specific points, but there's different ways that genetics shows up. Like you can have like the same gene showing up multiple times, and if you have more, it's called copy number variant. So you can have a single copy of a gene and not be very good at processing carbohydrates, or you can have multiple copies and that doesn't show up. And there's a lot of stuff that doesn't show up with SNP genetic testing. That's by far the most common testing commercially available right now.
Yeah, and a really good example of this is Helen was just talking about this copy number variant. So you could have like 7 copies of a certain gene in your genome, and one of them could have a mutation, and 6 of them don't. So then it gets complicated. Biology is complicated.
Yeah. And I think one of the things that you stressed throughout this book, and Krista also mentioned at the top, is just how much of an infancy we are in with all of this stuff. And I think you used a really good analogy within the book of like looking into a toy store through a dusty window, where you can kind of crane your neck and you can see different things. And there's so much potential there. But at this stage, we're just not at the place where we really understand everything, or we're able to do a hell of a lot with that information. So one other thing I wanted to maybe start with is just some different terms. And some of these will be useful because I imagine they'll come up during our discussion at other points, while others I think are just interesting within themselves and can start to explain certain characteristics about how genetics work, or how the body works. I'm going to direct these questions at Helen, but if anyone else wants to add anything, then you can. So the terms genotype and phenotype, so what do they mean?
Well, I'll use this. So phenotype is the trait that you actually see, the outcome of the genes. And so the example that they tend to use in high school is eye color. It's more complicated than we thought, which is kind of the theme. So with eye color, there's brown, there's blue eyes, there's green eyes, hazel eyes. So you can have, those are phenotypes. So that's the outcome of the underlying genetics, and genotype is the genes, the actual, what is the code for the variant for the phenotype. So phenotype is what you're getting at the end of the end product, and genotype is the plans, for lack of a better analogy. So that's the major difference.
Okay, and then the term epigenetics. What does this mean?
Well, epigenetics, literally it translates, epi is above, and genes is genetics. The genetics is obviously the gene, so it's above the gene. So what it is actually a modification of the DNA without actually changing the letters, the coding. So I use the analogy of if you have a book, you're not changing any of the words in the book or any of the letters in the book, but you start paper clipping certain chapters together, so you can't quite get to them. And that's what epigenetics does, is it modifies the DNA so that your body can't get to certain coding regions. And so and that threw a big wrench into genetics, because it's like, oh, we're getting this coding, okay, we'll figure this all out. And it's like, it's more complicated. Right. So, so epigenetics is another layer, and this can be modified both due to environment. So you can have, we're going to be discussed it in the text and then in the book, where there's identical twins that are, one is obese and one is lean, and they're genetically exactly the same. And the difference is due to epigenetics. So there's a modification of their genes where they're responding to their environment differently.
Okay. And so, when I was reading through, I was just trying to decipher the difference between phenotype and epigenetics. Because with phenotype, is there any environmental impact on that?
Yeah, well, epigenetics, so epigenetics is kind of the in-between, you could say. So you have your genes, and then you have epigenetics, which is like whether or not this gene and this protein is being made, and when it's being made, and that then dictates your phenotype.
Okay.
So it's again, there's different, it's just a different level of regulation. So yeah, so the outcome at the end, and this is my take, and I started in physiology, so I'm kind of biased. I think at the end, the physiology, which is the phenotype, trumps everything, because it's what the end outcome is. Whatever it happened beforehand is like, where are we at now, gives you a better indication, right? Okay. So if you technically don't have the gene to hate cilantro and you hate cilantro, it's like, what does it matter? You know, so, right? So, yeah.
Okay. And then like polymorphism. So that's another one that comes up a lot. What does that mean?
So polymorphisms are different variations on genes. So you have a given gene that makes a protein, but you can have different variations, and you call it, you call it flavors. I guess people could kind of understand that. So you have, let's say, ice cream. You can have vanilla, or you can have chocolate, or you can have strawberry. So these are different variations of ice cream. And you have the same thing with genes. And the variations are essentially the same thing, but they function a little bit differently, right? So chocolate and ice cream is the same and yet different from vanilla ice cream, right? So that's the genetic variance, but it's essentially the same protein with some minor differences.
Okay.
Or it could be major differences, but that's essentially it.
Okay. And then heterozygous and homozygous.
Going back to, I guess, the eye color thing, because it makes things easier. So homozygous is that you have, and I know maybe Alaina could jump in, because I'm like trying to explain it without over-explaining it, is that homozygous is you get DNA from your mom, you get DNA from your dad, right? Okay. So you have basically two copies of every gene. And if they're exactly the same variant, then you're homozygous. So homo being same. If you have different versions or different variants from your mom and your dad, then you're heterozygous. So for you, if you have brown eyes, which is the phenotype, you could have both homozygous or heterozygous because of the way the genes function. So you can't tell. But if you're blue-eyed, then you definitely have homozygous for blue eyes. So it's basically, you have two copies of every gene. If they're the same, you're homozygous. If they're different, you're heterozygous.
Okay. And then monogenic and polygenic.
So monogenic is the idea that there's one gene that's responsible for the outcome, the phenotype. So again, eye color, they thought was just one gene, and that's like more complicated. So then now it's polygenic, sorry. And so, yeah, so basically mono is single, poly is more than, and the vast majority are our traits or phenotypes are polygenic.
Okay. And I think this is something I do want to highlight, because I think often in the media, when people talk about this, or often people's thoughts around genetics is that there is an addiction gene or an obesity gene, or like there's this one gene that is having an impact and just drives everything else. Whereas now what we're seeing is that it's potentially tens, hundreds, thousands that are all working together to make certain changes.
And what it's interesting is that we kind of figured out a lot of the genes that were like monogenic, because if there was a disease, that they figured this out before genetics came along. They just use family patterning, and like, well, basically it was figured out through ancestry, and they could follow the genes, and they couldn't locate it, and they didn't know exactly where it was, but they knew it existed. So the simpler ones were kind of discovered or well known prior to genetic testing that we have today.
Okay, is Huntington's disease, is that one of the ones that's pretty much like that?
Well, Huntington's, if I remember, actually, Alaina, do you remember? I think it's a little more complicated. That it's like multiple, I think, copies, isn't it? I
can't remember exactly, but like Huntington's and cystic fibrosis are two of the genes where, like, if you have this variant, you're super likely to, if you have like Huntington's, you're probably going to die from Huntington's.
And they knew that before, before they got to do it, they're like, we have a pretty good idea that this is because of a single gene or a single, they call it alleles before they'd understood genes. So they had basically a unit of heritability is what they were calling it. So they figured out that it was heritable and it was from a specific, likely one source.
Okay. Another one is haplogroups, if I'm pronouncing that correctly.
Yeah. So haplogroups is an interesting thing that the genes are clustered together, and then they tend to move down at the heredity together. They're kind of like hanging out together, so they'll move along genetically together from grandparent to parent to child, and that's more of something to keep in mind when when you get tested, because a lot of times they don't test the specific SNP, but they're kind of extrapolating to other regions. So I don't know if we want to go any deeper than that, because it's more of it's an interesting, technical issue. Okay. As far as how things are being determined and tested.
Okay. And so does this link into the ancestry part of genetic testing or not?
Yeah, well, Alaina, I think, has actually a pretty good background in that part of it.
Yeah, so I mean, the answer to that is yes, right? Like, when a mutation shows up, if it shows up in cells that get passed on to your kids, like sperm and egg cells, then it enters the germline, right? Like that's the kind of genetic lineage of whatever species that you're talking about. And so these groups of things tend to happen, like, and because I would say it's uncommon for a single, like a SNP, to get mutated in exactly the same location once in northern China and once in South America. Like, you can probably assume that if you see this variant and it shows up here, then it's much more likely in this population of people who live in the Andes than it is in the people who live in northern China. And so if you see this variant in a person, then you can theoretically trace back to where this variant happened. And so when they talk about like maternal haplogroups, what they're talking about is like the genes passed on from the mother, in particular in the mitochondria. That variant is belongs to a certain ancestor. And this certain ancestor is an ancestor of all of the people who have this variant. And so now I can say, oh, this thing that shows up in people in northern China, this person has some sort of northern Chinese ancestry, is likely to have some sort of northern Chinese ancestry.
Okay. And one of the things that you stressed in the book as well was just how much of the genetic testing or the research often is done on white Europeans, and the fact that your ethnicity or your background does have an impact, so that your genetics can behave differently because of your lineage than someone else who is, as you said, like from an Asian background or whatever, and that it's also a lot messier than just that. Because I mean, I did my own genetic testing, and I'm mostly Northern European, I think Irish. But then there's all these other things that are then thrown in there as well into the mix, and so it starts to be like, well, how much should I be relying on that bit of research? Because is it matching up exactly to my heritage?
And the other aspect of it that's interesting is that if you have a sibling and they got tested, they would come up with different percentages because they, so it's a question of, I always found it fascinating. It's like genetic ancestry isn't the only ancestry that you have, right? So that's the other thing to keep in mind. But it is really a really fascinating aspect of it.
Is that also down to the labs? I think I was hearing someone would talk about this on a show where they got their genetics done, 6 years ago with one lab, and then when they got it done with a different lab, their ancestry looked different. And when they started to investigate why, it looked like more of a certain group of people were coming through that lab, and so that was why it skewed the data. I don't know if that's correct or not.
Yeah, I could take this one. I mean, that is definitely true. Like, we know a lot more about you, about what your genetics mean if you're a white European, and so one of the things that's happened is that these companies like 23andMe, for example, are building their own data set. And so if they have a lot of white Europeans showing up, then they're going to have like a certain set of assumptions, which is why there are efforts like the 1,000 Genomes, I think, is one of them, or the National Geographic's Genographic project, that are trying to get samples from all of these different parts of the world. Because once you do that, you're like, oh, I mean, like genetics, really, the study of genetics at its foundation is like a statistical study, So if I have fewer data points, then I have a larger uncertainty. And the more data points I get, the narrower my uncertainty becomes. And I think that's a really important point to talk about with genetics. Biology in general is like statistical. Like we think of it as like something that's squishy, that you can touch and like sometimes liquid squirts out and stuff like that. But like at its root, the way that biology works and trying to predict how biology works is a statistical exercise for us.
So that, nicely then follows on to something else that I wanted to touch on, which is the concepts of like absolute risk, relative risk, and odds ratio, which are really important when you start to look at genetic testing, because genetics aren't set in stone. It's looking at the risk. So do you want to just, Alaina, talk about what those different terms mean so that people can understand it?
Yeah, so those aren't specifically genetics terms. Like you hear those a lot in epidemiology, right? So epidemiology is like the study of populations and how things move through populations. So the absolute risk is like in a given population, like out of 100,000 people in North America, 40 of them will get colon cancer. Like statistically, 40 out of 100,000 people in North America are going to get colon cancer. And that's like absolute risk. And in an odds ratio, well, relative risk is a kind of odds ratio. So like relative risk is the likelihood that something in a certain group is going to show up in a certain group compared to another group. And like the classic example of this is cancer. So if you're a smoker, you're like 10, 20, 30 times more likely to get lung cancer than a non-smoker. So the relative risk of smokers versus non-smokers to get lung cancer is like 20 times or 30 times. I don't know what the exact number is. And then you can, odds ratio is like this odds that these things will happen in different groups. And relative risk is like just an example of an odds ratio. But you can extend this to genetics, right? Like your absolute risk could be like out of 10,000 people, 2,000 of them have sticky earwax. But relative risk would be like people with a genetic letter A in this location are 7 times more likely to have sticky earwax than people who have the letter C in this location. And that kind of gets at what you were like it gets at lots of different things, but that's really what those things mean. And that's why with these polygenic traits, like you can't say, if I have this, like we even actually did our own little experiment, and we had a whole bunch of genotypes from people at PN, and we're like, okay, well, let's look at this well-known I hate cilantro gene. And so we actually went through and then we asked people, hey, you hate cilantro? And it turns out that it wasn't very predictive. Like in our small subset of people who work for precision nutrition, like there was, I don't remember the exact correlation, Krista, do you remember? But it's like, it's not like that. It doesn't work like that. So I can't. Loose. Yeah.
Very loose.
And the other aspect of this is that I think people like, oh, well, I got genetically tested and I have like an X times increased risk of whatever, let's say cancer. And it's like, well, what's the absolute risk? And it's still infinite, like it's ridiculously low. And they don't see like their daily like activities and how that impacts the outcome. So people do genetic testing like, oh, so now I'm like done for, I can't go on. But meanwhile, it's like, well, if you ate more vegetables, that's going to have a bigger impact on your outcomes, right? And so it's, I think, not only how much we know clinically as far as what your genetic variants impact your health, but how much does it really impact? And people are very bad at really assessing risk. And also I think, when they say, well, this gene will like cause cancer or whatever, it's like, well, what are the odds? Like, is it increasing? It's not going to cause cancer. It's going to increase your chance. Like the gene doesn't go out. Like this is not its job. Its job is not, there isn't a gene that its job like in the genome is like, my job is to give you cancer. Like it doesn't work that way, but it increases your likelihood of this outcome. And so people are, and I think a lot of times there's a misunderstanding of what the interpretation is, even if it's like a sound interpretation.
Yeah.
Yeah, and I think there are a few examples of that and stuff that showed up in 23andMe. Like they're really funny about health traits, like for good reason. And so like you might have a slightly increased risk of breast cancer, or Alzheimer's is the one that I'm sort of like emotionally attached to, right? Like I have like a slightly increased risk of Alzheimer's, which is not like a major realization because I have several like older family members who have had Alzheimer's. Like I'm not surprised by this, but I know that what I can do if I have this is keep my mind active and eat well and exercise and get good sleep and do all of these things that like much more likely to reduce my risk in this stuff. Like that's just kind of the environmental stuff that Helen is talking about.
And you should be doing that anyway. Like, which ends up being the caveat on almost everything. Is like, well, and you should be probably doing that anyway. It's basically a lot of the genetic testing results. It's like, you should sleep more. Like, yeah, you should be sleeping from 7 to 9 hours a day. Well, yeah, I probably should be doing that anyway. So.
Yeah. And so let's then talk about specifics in terms of different labs. And I know you go through this in the book, but I think it's something that people would really want to know about. So if someone's thinking, hey, I would like to get some genetic testing done, what would your recommendations be? Who should they be getting this done with? And I'll direct this to Helen.
I guess it depends why they're doing it. Okay. I think if I'm concerned that I am at risk for breast cancer, or I have an issue like Huntington's, or concern about I have spina bifida in my family, I'm concerned about and I want to have children, then I would speak with your medical professional, your doctor. Probably a genetic counselor should be involved in that case, where there's it is fairly it's well established, and the outcome is pretty. Can have a major impact, or there's something that you can, it's more of a medical diagnosis. If you're doing it for entertainment, you kind of can go anywhere. You can go to, if you're interested in ancestry, depends, if there's a lot of different ones that go into ancestry. If you're looking, and how much you want to pay, and a lot of them, it depends, the restrictions that they have. And Alaina's done a lot more as far as testing for a combination of entertainment and trying to get a prescriptive, that's more of interpretation. But if you're talking about just purely testing, Alaina has been tested multiple times. She probably has better experiences on that. I could do the testing for you, but I haven't got other places to get it done.
Okay, so Alaina, do you want to chime in?
Yeah. So, here's the thing about most different companies that offer testing is that they all use one of 2, 3, 4 companies to actually do the testing, right? So 23andMe, I think they've just released a new version. I don't know anything about that, but they're the chips, the little microarrays that they use to do this testing are made by a company called Illumina. And so if you go to Gene by Gene, who does more clinical testing, they use Illumina chips. And so one of the things that we did, you had talked about how someone had done their testing at one thing and then did their testing a few years later, and it could be different. So that actually happens all the time, but the variation is very small and it's well understood. So we did, I have had, I don't know how many different, I had my genotype done on 3 different 23, I spit in a tube 3 different times for 23andMe. I spit in a tube once, and then Helen and I actually took a blood sample of mine, and we prepared our own DNA, and we sent both of those different things, saliva and blood, to the same company. And then I've had my whole genome sequenced twice. So from beginning to end, I know all of everything in my genome. I think really what Helen is getting at is we wrote a 400-page book that says, we don't know much yet. That's really what I would
say that technically, I think you're fairly confident going with almost any company that's not you're in a shady corner of the internet.
Yeah. Any company that's reputable, that follows standardized laboratory practices.
It's going to be the same.
Yeah.
So technically, like, you're going to get the same, like what's the letter sequence, or what SNP variations do you have, is probably going to be the same. The issue becomes the interpretation of that. Like, what does that mean? And that I would say is, again, if you're doing it for entertainment and you're not, but a lot of people don't want to know more than the very essentials that they want to know, like a very narrow thing. So so then it becomes another level where, is there more clinical evidence for supporting the interpretation, not the actual testing? So it's not the testing so much that's the issue. It's like, what does this mean? That's the issue. And by and large, we know a lot less than most companies are trying to sell you.
Yeah. It drives me absolutely bananas to see people advertising, we're going to, test 30,000 locations on your genome and tell you what diet you need to do. Like, that's, I don't know, that like offends me on a moral level, because they're making a claim they can't support.
Okay. Just because we know so little, and then also the fact that relative risk is only going to tell you so much in terms of pointing you in a certain direction.
Yeah, and a lot of that isn't even going to show up. Like, I can have a certain genetic code, but my epigenetics could, epigenetic modifications could turn it off, and that gene never gets transcribed.
Yeah.
Okay. So, I mean, one of the other things that you do really hammer home, and this is kind of the same thing, is the problem within research, which is known as like the replication crisis, or that's what it's been labeled recently, where there's research done and it produces a particular outcome, and then when that same research is repeated, they get a different outcome, so they aren't able to replicate the result. And there's many reasons why this can be happening, ranging from like the benign to outright fraud. And then there's certain areas within science where they'd come under more scrutiny because this appears to be more common. And research around genetics is one such area. So there's a lot of false positives that seem to happen. And so this is something that is covered in the book. So I'd love to hear you talk about this, and also reference maybe the example in the book around chemotherapy. I don't know, Krista, do you want to chat about this, or am I better giving it to someone else?
Well, I'll kick it off and give people the broad contours. I think we have to understand the context in which research is done, and Helen can speak to this as well. But as someone who's a former academic, I can certainly grasp and think about this. So most research is not sort of this wonderful, like, in the pursuit of science kind of research. It's based on funding. It's based on funding cycles. It's based on time. It's based on what the institution or the funding body deems as a priority. So as a researcher, you're not just free to explore whatever question interests you most of the time. You're bound by particular limitations or agendas of the institution and organization that you're working with. And academics are under a lot of pressure to publish things, to find things, to discover things, to move stuff through the lab or through the research agency. And so what we often see is that research is generated that may not be well replicated, that may not be well substantiated, that may go in a certain direction while missing other directions. And it's not that there's nefarious agendas or anything like that. There's just a lot of human factors in research. And so one of the most crucial questions with good research is, can we replicate it? So can some other person working somewhere else, or some other lab, do the same study, the exact same study, and get the same results? And that's how we know things are true. So, with gravity, we could all go outside, drop the orange off the Leaning Tower of Pisa, just like Galileo did. I think it was an orange, right? And we can all replicate gravity independently. And so as we get more and more into having consequences for genetic testing, we need to really push for replicability. So the case that you're talking about was, now this happened about over 10 years ago. So a lot of the regulations around this have changed, but one group of researchers claimed that they had found a way to personalize chemotherapy based on genetic testing. So this sounds really awesome, right? Like, who wouldn't want that? And in fact, another group of researchers sort of went ahead with, oh yeah, that's great. Let's establish clinical treatment guidelines based on these findings. But then another group of researchers was like, I don't know if this was really, and best possible interpretation was something like someone was rushing, or like there was some data error or something like that. Worst possible interpretation is that there was outright fraud. Who knows what the true story was, but it's a very cautionary tale about the idea that we have to make sure anything we have is well replicated before we start basing like clinical treatment guidelines. So before because a clinical treatment guideline results in someone going for chemotherapy, getting a particular type of drug. So someone is sitting in a chair getting drugs based on what this researcher found. So it has very, very real consequences sometimes. I don't know, Helen, you probably have thoughts on research in the institution.
It lends to this idea of like, you can't read a study, this new study came out and it revolutionized, whatever heck it came before. And it's like, probably not. It's like, for me, I've gotten to the point where I was like, something new comes out. Okay, that's interesting. Make a little note of it. And if then I want to see it again. I want to see it repeated by the same lab. I want to see it repeated by another lab that's not associated at all with this lab. And then it being built on, whereas if this is true, then the next thing should also be supported, the next study. And so, for me, I don't really feel comfortable until it's been 5 years out and I see replication from different places. And not necessarily exactly the same study, because that is really hard to publish, but something that builds on it and comes out. Because I've, during my grad studies, there was labs that they would publish in Nature or Science, and then they'd never publish again in that area of that. There was no follow-up, which was big red flag that this was not true. Or only one lab has ever published about this specific topic or about this genetic outcome. Another big red flag. It's what are you doing exactly? And again, it's a lot of it is patience, and it's not, if you wait, the truth will kind of shake out. It's just that I think that we want to know now. And people are like, so science is wrong. It's no, you kind of have to wait and let it kind of move through its process. And the longer it's been around and the more it's been vetted, the more it's been tested, then the more you have more confidence in it. And that's what people need to understand. The longer something's been around and been supported, it's more likely to be true.
And in terms of genetic testing, if you were to be guessing, and I know this is just complete speculation, and this, Helen or Alaina, you can answer this, when do you think we're going to be at a stage where you can be doing what they were hoping to say with the chemotherapy? So we can look at your genetics and we can say, okay, these are the drugs that are going to be better for you based on that. Or that genetic testing becomes a fairly big centerpiece as part of our medical or health care system, if that's even going to be a thing.
Yeah, I think, it's interesting. If you look at chemistry, chemistry is because if we've gotten to the point where we kind of understand, we're pretty confident we can predict stuff in chemistry and how chemistry works. If you put this and this together, then this is what's going to happen, and you get all the percentages. And it's nice and clean because you can repeat it really easily. It's very consistent. And then so you bring in biology is messy because you have different variations of people and they don't respond the same way. And then they refuse to live in a lab for their entire lives so you can test them and stuff. So it gets, I would say, 50 to 100 years before we get, and not to say that you're not going to find pockets of things where we can do, and I think, tumor testing of cancerous tumors and chemotherapy may be one of the easier hurdles. But, nutrition, being meaningful nutrition prescription based on genetics, I'd say 50 to 100 years. I know there's people in 5 years. I'm 5 years? Gee, I did grad school. 5 years is nothing. You didn't get anything done. I know, Alaina, what's your prediction?
Yeah, it's on the order, I don't know that I would even put a number to it, but it's on the order of decades. And I also don't think that genes will, I do think the cost of genetic testing, the cost of sequencing has decreased dramatically. Whatever it was, 2000 or 2001, when we finally had the whole human genome sequence, we were oh my God, this is amazing. We've learned everything. And then it's we don't actually know anything. And so that was 18 years ago, right? And we're still we're a little bit closer. And I don't think that, I do think we're going to get to a point where it's cheap enough that if it's $100, then we'll probably like sequence every kid's genome and that'll become part of your medical record, right? Like, I don't think it's ever going to be the center of healthcare, though. I think what is probably going to become much more important is like what genes are transcribed and like how, like what proteins are created at any given time. And it's different in different tissues, right? That's why lung cells are different than heart cells. So it's just much more complicated. And I think we had like this Pollyanna idea of what it was going to be, and it's not. Yeah, and we're always looking for like these simple, straightforward answers, like yes or no predictions, and biology doesn't work like that.
Okay. So look, what I want to do is talk about specific areas of health and some of the genetics that we know in this area. And what I really liked that you did in the book, and you made reference to this already, is that you all got your genetics tested, and then there are other people at PN, and I think other people's parents at PN. So I think there was 12 or 14 or 16 people who did genetic testing. And so what you did as part of the book was look at what the research would predict based on different SNPs, and then looking at what happened when you surveyed your group and then looked at their genetics and how that matched up to what should have been predicted. And so I thought quite a lot about this section and how I could make it useful for people. And rather than asking you about specific SNPs, I thought I'd just allow you to focus on the things that you believe are most relevant in certain areas. And I'm going to direct the questions at Helen, but again, others, Alaina, if you want to chime in, then please do so. So if we're thinking about the section on food preferences and genetics, what are some of the genetics that you want to highlight for people, or things that you think are interesting in this area?
I think it's interesting. I think, people who have children maybe, and it's like the kid is like not eating broccoli at all, and you're like, and it's like, is it, and so I think more of it knowing that it exists versus I'm going to genetically test my kid to see if they're going to, they have a polymorph or a SNP where they're more sensitive to the bitter taste in broccoli and cabbage and whatnot. So it's, I think, awareness that people are they vary in this way of how they taste sweet and and it's not like you're wrong. This tastes terrible, right? Right. So there's a website. It's called IHateCilantro.com, where there's these people who do have this hatred towards cilantro, and seems to be a genetic basis for it. And I'm assuming there's going to be a lot of people in that group that have, the polymorphism for hating cilantro, but not necessarily 100%, because who knows? They maybe had a bad experience when they were a child with cilantro, and it has nothing to do with genetics, right? So I think as weird as it sounds, it's not the testing per se, but being aware. I think there was, what was the one? I can't remember what gene it was as far as, fructose, fructose, being able to metabolize fructose, that, people are trying to, is fructose good for you? Is high fructose sucrose bad for you? And this and that. And it's, well, probably how much you're taking is going to be a bigger factor. But if you have this, the same way if you're like lactose intolerant, you have a mutation for lactase. Well, you can kind of figure it out a lot of times, whereas like I drank milk and now I feel terrible. What should I do? Well, it's like the old joke goes, well, stop doing that. Right. So a lot of the preferences, the food preferences is, there could be underlying genetic reasons. And that's something to keep in mind. And I think generally with children, I think parents have to be kind of aware of that. The other part of it is that there's an environmental component. As you get older, you're not as sensitive to things regardless of what your genetics are. And so if you're a smoker, then that's going to make even less of a difference, right? So I think the context of it and the awareness more than should I go out and test my children to see if they don't have the variation for not liking broccoli, then God damn it, they're going to eat all their broccoli, right? So, and so it's interesting. I don't know. I find it more of an entertainment value for food preferences rather than people ask me, well, what should I do if I have this mutation or this polymorphism? I'm like, so. I don't know. Does it matter? Does it matter if you genetically don't like broccoli, or if it's like some environmental thing that happened to you, or you don't like the texture of it and we haven't figured out the genetics for that?
Yeah, and it was interesting when I read through that section, because I've got a young son, Ramsey, who's 7 months old, and we've just started feeding him, and we're now doing baby-led weaning, which is what's recommended now over here. And I'm wondering how much of a difference that will make from an environmental factor versus what would have happened 5 years ago, 10 years ago, when the recommendations were, okay, babies get the same pureed food for the first 1 month, or whatever the recommendations were. So having that variety from a very young age, what that does in terms of the genetics and whether they really shine through or the environment just washes over it.
Yeah, so probably at the end it ends up being a both, right? It ends up being a mix, right, at the interaction, and then now the microbiome throws in, and that makes it even more complicated. I think a lot of it is kind of paying attention, and when you pay attention to what's going on, people say, I can get away with 5 hours of sleep a night, and then you start asking them questions about how well they're functioning, and you're like, are you really doing well with 5 hours of sleep a night? So if you're just paying attention, they're, as far as I know, I don't think they've narrowed down the gene for people who can sleep longer, less, or longer. But at the end of it is, like, I think if you look at how you're functioning, right? So if you give your son, broccoli, and he refuses, you're not going to go like, you know what? I think we're going to do some 23andMe testing and we can figure this out, or cilantro. So, that's kind of the takeaway, I guess, for a lot of this is if you're paying attention. And there was I can't remember the conference where they actually genetically tested. They did sequence genetic experts. So like, we sequenced your genome. And so now that we have this, what do you think? Like, what can you tell us? What do you think it tells us about you? And then they pretty much felt like, you talk to us for 5 minutes, you probably have a better sense of us versus, what you can get out of the genetic sequencing at this point. And so that like, and I think that's what people are missing, is that this idea that you're going to get some information that you can't get with genetic testing. So with genetic testing, you're getting something not like something earth-shattering. You're getting a little more piece to the puzzle.
Okay. Mm-hmm. And Krista, I mean, I wonder for you, if I'm remembering correctly, are you still coaching clients, or you're now more like overseeing people who coach clients?
Yeah, no, I coach clients.
Okay. So how much do you use genetic testing as part of that? Would it impact your suggestions with clients? And the same with like other coaches at PN, like what are they doing with genetic testing?
I think Helen makes a really good point. You can just talk to someone for 5 minutes and find out more information. And so for me, there are very few at this point genetic tests that are truly determinative of things like food preferences. There are other things like how much do you like fatty tastes or sweetness. But again, you can learn that in 2 minutes. And the thing about food preferences that are really interesting in the context of coaching is they're so malleable. Like, there are very few genetically determined food preferences that are truly unmalleable. I think the bitter taste one might be one where, some things just taste so bitter to you that it's hard to overcome. But I think even then you could still learn to prepare things in a way that like cuts down the aversiveness for you a little bit. But food preferences are such a wonderful example because they're so malleable. They're so incredibly changeable, even in a relatively short time. Even stuff like we have a 2-week no caloric beverages habit. And the way we tell people about it is to say, okay, play with having calorie-free drinks for like 2 weeks. You don't have to have calorie-free drinks forever, but just try it. And at the end of 2 weeks, people have taken sugar out of their coffee. They've taken cream out of their coffee. They've switched alcoholic drinks. Like they've made huge changes in only 2 weeks. So they start off day one, they're like, Mom, my coffee tastes like dirt. And then 14 days in, they're like, it's not so bad. Maybe I'll try espresso next. And so it is fascinating to me. And also as someone who travels internationally, to see how food preferences change worldwide, and to see babies who we think, oh no, we have to, babies don't like things that it tastes too strong. I've seen babies eat kimchi, raw fish eggs, hot peppers. I've seen babies eat all kinds of stuff. That makes that sound really bad, doesn't it? But But we have this idea, I think, that is very North American, that babies and small children have very limited taste preferences. But culturally speaking, if you travel around the world, you see that's very much not the case. And to bring it back to coaching, again, people have changed their preferences in a few weeks, a few months, and certainly over a year. So like almost no food preference is so determinative that it's going to shape your entire life. Now, it does seem like there are people who have some kind of sensory processing issues, who are the true picky eaters. But this seems to go along with a whole other suite of kind of sensory disturbances. So it's hard to say that you can't really say there's like a gene for picky eating. It's not like that. So the food preferences thing, I'm glad that you picked that as a topic, because I think that's one of the best examples of how irrelevant genetic testing would be and how useful plain old everyday coaching would be. And I think there's a line in the book, this was around exercise. There's very little that genetic testing can tell us that a coach standing in the field with a clipboard could not see. Like any good, even a high school football coach can tell you, this kid's going to be good, this kid's going to be good, this kid's going to suck. Like it's obvious. So really, there's almost nothing that you're going to get. And I think either Helen or Alaina said, it's fun, it's cool. You might discover why you hate cilantro, but other than that, not much.
Yeah. And with the exercise thing, when I looked through my 23andMe, it told me that I should have the muscles of like an elite person who like power lifts or kind of puts on a lot of muscle, which is the complete opposite of me. It told me I should not do endurance. I did a marathon after doing 8 training runs. Like a lot of when I got my genetic testing back, I was like, did I get the wrong person's results? Because I mean, it said that I should be at a higher body weight than average. And I, for my whole life, have struggled to put weight on. And so, yeah, I do think there's things that point towards what might happen, but yeah, it's definitely not set in stone.
Well, and I think the exercise is another great example, because so you might say, well, what about for elite athletes? So, okay, forget about regular people. Maybe it doesn't matter for regular people, but what about elite athletes? What about Olympians where a thousandth of a second can make a difference? Well, even so, those Olympians have to get out of bed in the morning and go and train. So there are thousands and thousands of hours in an Olympic performance. Genetics gets you in the door to be an Olympic athlete. But after that, it's everything else. It's psychology, it's training, it's funding, it's all this other stuff. So even at like the most elite levels of human performance, anyone who specializes in human performance will tell you that training and practice and all that kind of stuff is a million times more important. I wonder too if part of it is our paradigm about how genetics works, which is just that if you're good at something, it means that you're talented or it means that you're genetically gifted. But that's not how performance works. Genetics will merely give you the baseline. Like at 5 feet tall, I am not getting in the door to play at the NBA, right? It's just not going to happen. I actually always kind of feel bad for people who are like 8 feet tall and no good at basketball, because then it's like, well, I really wasted a gift here. But so yeah, okay, so you have to have a certain genetic set of features to dance ballet or play basketball. But past that, it gives you nothing. So even for elite performance, we can't really use it.
So with all the different areas that you looked at, so you looked at like metabolism, you looked at exercise performance, a number of different categories. Were there any categories and were there any things that you discovered as part of that you thought, okay, this is really useful information that I couldn't get somewhere else?
Not that I couldn't get somewhere else, and I'd be curious, Helen, what you think. For me, the most intriguing part was the ancestry and heredity data, because I think that so much of our recommendations are based on a very restricted population of white North Americans of European descent. And so seeing a lot of the ancestry and heredity data and really appreciating the vast diversity of human beings was very, very useful for me to really to see it all laid out and go, whoa, like even if we're talking about, we talked about before about haplogroups, even if we cluster people into groups, there are still several giant groups that people are in, never mind drilling down into the detail level. Personally, like that was the most useful thing. And also to trace, to see like, oh, actually for me, like there is probably some East Asian genetic material in there because my ancestors lived in Eastern Europe and there was lots of kind of back and forth. But other than that, I think there's not much you could get elsewhere that you couldn't get elsewhere. I don't know. Helen, Alaina, do you agree?
Well, like as far as genetic testing, I think there's some stuff that it would be more definitive. And again, those are the things that you would go to a genetic counselor for.
Yeah.
With the exception of maybe like if you have a history of spina bifida, family history of spina bifida, it might not be something that would show up in genetic. You can get genetic testing to see if you do carry a specific version of the gene that metabolizes folate. And so if you're a woman that wants to get pregnant, then that you might want to get tested if you have family history, so that you would then going forward, you would want to make sure you're supplementing with folate and a specific type of folate, so that you reduce the risk of your child getting spina bifida. And then that's something that's actionable and has a massive outcome, and is one of the few things that is very cut and dry as far as risk. And but I'm trying to think outside of that. There's other things, but again, it's very phenotypic, where it's like you can just look at the outcome of you doing X or Y, or it's not actionable. It's like, yay, you have decreased risk of this. There's not much we can do, So that's about the only thing I think of. Yeah.
Yeah. So I guess my interpretation is like there are definitely there's limited clinical usefulness, but the thing that I marvel about ancestry is that we have these hypotheses about how the human race spread across the world, right? And using these genetic tools and tracking different populations, we can actually see where that happened. Like if you're of European descent, you're more likely to have some Neanderthal DNA, because there were events where Neanderthals, Homo neanderthalis, and Homo sapiens interbred. And so you see that there, but you don't see that in Australian Aboriginals because they had migrated before that happened. And using these things, like now we're starting to mix up like the sort of massive human genome as we like different groups breed together, which is generally a good thing. Like you want more genetic diversity rather than less. But I don't know, I feel that's a very magical experience for me, going like, wow, we can see in our genetic code that this group left the Middle East 60,000 years ago. And we can follow them all the way along the coast of Southern Asia. And we can tell when they crossed from Indonesia over to Australia. Like we can see all of these things. That is fascinating to me. That is how we know, like how we have more evidence that people, migrated from China up into northern Russia and across the Bering Strait and down into North America. And you can see where different populations stopped along the way. That's amazing. That's amazing that we had the ability, like anthropologists have done all of this research, and then we can see it in the genetic code. It's miraculous to me. It's what an amazing thing science is.
So something, and I want to keep hearing from you, Alaina, something that didn't come up in the book, but I want you to cover is CRISPR and then CRISPR-Cas9. So are you able to talk about what this is?
Yeah, I love this futuristic stuff. So CRISPR is something, it stands for clustered, regularly interspersed short palindromic repeats. And what these are in bacteria, there are these little stretches of DNA that are, about 20 bases long, and they appear in different places in the genome. And like we were doing bioinformatics on this back in the 90s, and people are like, wow, this is really interesting. What is this? And so it turns out that bacteria have an immune system too. And so bacteria have to defend against viruses, bacteriophages that like infect bacteria. And so what this CRISPR system does is it effectively patrols the cell looking for alien DNA. like DNA from viruses. And when it finds that DNA, it makes a copy of it and sticks it in the bacteria's genome. And then it's always looking like it's going through its library of viral DNA, and it's looking for this in the cell. And when it gets it, when it finds it, the Cas9 is what's called a nuclease. It's a protein that like attaches itself to DNA. So it finds this little stretch of viral DNA that it recognizes, and it attaches there and snips it. And so when it snips it, whatever that gene was can't get made anymore. So like a virus might insert something in there that, you know, makes more viral proteins, like the surface proteins on a viral capsid. And so if I snip that, then the viral capsid can't form, and whatever DNA or RNA goes inside that virus isn't going to affect anything else because the container doesn't work. So this is just like a basic system that bacteria have evolved to do this, and it's in many different kinds of bacteria. And so when researchers found this, they were like, oh, bacteria have DNA, mice have DNA, humans have DNA. We can use this same system to make edits to non-bacterial genomes. And so what they did was, and now I can match up any DNA, right? So like if I know that there's a cystic fibrosis variant, I can match the DNA that's there, and I can use other processes in our cells to like snip that part of the DNA and then replace it with something else. Like I can insert a template that says, okay, there's this crappy version, but don't do that. What I want you to do is take this good version that I've actually given you a template for and make that. And if I can infect, like using viral vectors, if I can infect lung cells and I can, you know, make this edit and like change the cystic fibrosis gene, then I can cure cystic fibrosis. And so it's an incredible tool. Like we've been able to do this for a long time. We've been able to like edit the genome. It's expensive and imprecise. And what CRISPR-Cas9 has done, and so Cas9, by the way, is that nuclease that does the clipping. So what this has done is this has let us make very specific targeted edits very cheaply. And it's like this, it might not be CRISPR-Cas9, like there are many other kinds of genetic editing tools. There's probably another one coming up that we don't know about. But this is the beginning of something where we're actually going to be able to change our genetics. We do that now, you know, but like it's so cheap and it's so easy to do. Like I guarantee you that there's a Nobel Prize coming for this discovery and the description and the use of it. I don't know when it's going to happen. Like, you know how the Nobel Committee is, it'll take forever, but this is super important. Like this is really, really important. This is happening. And it's an amazing thing. Like I'm not even a molecular biologist, and I've used CRISPR 3 times in the last year to do things in like synthetic biology in you know, plates of bacteria.
And I'm going to be the downer because so gene therapy is basically, you know, the application, you know, I think the end product where we can say like somebody has cystic fibrosis, and we can give them, you know, this, we can, you know, replace the faulty gene that's causing the disease, not causing the disease, but basically it's not functioning properly, which then leads to the disease. But ultimately the biggest issue, because we've had gene therapy or the different versions of editing genetic or genes for a long time, but the issue has always been trying to infect people with, you're trying to save people by infecting their body, and your body doesn't want to be infected. For a long time, that was a bad thing. And so you're trying to get around a very, very fundamental, like, self-defense mechanism and that's been the huge challenge. So, you know, CRISPR is allowing a new way, and it's a much cheaper way, and there's a lot of different things you can do. But in mature organisms, like if you do it prior to you know, embryonic, you could probably do CRISPR at the embryonic stage, and even then you might run into problems. But once you're born, I think it's still going to run into the issue of getting behind the defense mechanism.
So that's what they're talking about doing with CRISPR-Cas9. Is it that it's not at that embryonic stage? It's like you in your 30s decide that you want to change some stuff, and then you take this thing in.
Yeah. So, but I mean, like Helen makes a really good point. Like the immune system, our adaptive immune system has evolved over like millions of years, right, to do this kind of stuff. And so what we're having to do to like insert this in is we have to find something that doesn't cause a disease in people. Like adeno-associated virus is one of the viruses they kind of use, but we can use it to target certain cells. And then suddenly the immune system is going to be like, they don't care. Like the immune system has no value judgment on whether something is good or bad, right? It just recognizes,
like not self.
Yeah, something that's not self. Exactly. It's something that's shouldn't be there. And so like where you will, and I agree, like that's a problem that could be solved. It's why I don't think that CRISPR-Cas9 is like going to be the thing that like eliminates cystic fibrosis, the experience of cystic fibrosis. There's going to be something else that we don't know about yet that fixes these problems. Like maybe we can sort of moderate the immune system itself. Like who knows what that's going to be. But if I can edit the genome of like an egg or a sperm, like it'd be much harder to do with a sperm, I guess. But if I could edit the genome of an egg, then I can remove cystic fibrosis from the human population. Like theoretically, I mean, it becomes a technical problem. Like, how can I do this? But I can do that, you know? And so once I do that, that variation goes away. And if I can remove it from everybody, then I don't have to like tell people that they can't have babies like you would in eugenics, right? Like what I can do is, okay, well, you know, you and your partner want to have a baby and like, maybe there's this trait that I can fix. I can remove the risk of Huntington's from people who have Huntington's. Like, are you going to do that? Maybe. Like, I think any parent is going to want the best for their kid, you know? And then you get into questions of like the ethical questions. So that's going to be accessible to people who are wealthy, you know, people who can afford testing and who can afford this modification. And you already have a stratification of society. Now these wealthy people start modifying their kids. And like right now we can imagine these like simple monogenic traits that we can change. But what happens when we actually do understand the polygenic traits and we can make several edits? And then suddenly rich kids living in suburban California are like incredible athletes, or their IQ points tend to be an average of 10% higher than kids who grow up in, rural Scotland. Like now you have a real imbalance. And I think that there are a lot of these ethical issues that we should be considering. And we're not, just because we're so technically far away from making this happen. But this is the point. I really believe that in 100 years, we're going to look back on this and go, this is the point where we should have started having this conversation and talking about like what the effects of this are. Because the other problem is that like we don't actually know. Like we might know that this variant increases the risk of cystic fibrosis, but what if it does something else? And we just don't know. Like we don't know enough to make these decisions.
Well, that's the thing too. Is I think a lot of variants. It's not like it was like this was a good variant and this was a bad variant of a specific gene. It was like this was a good variant in this situation and this was a good variant in another situation. So it's like sickle cell anemia is a good example where, so what ends up happening is what happens is red blood cells are misshapen. They're basically in the shape of a sickle. And this is very advantageous in places with malaria, but you don't want to be homozygous because then you have you have a lot of issues with not actually being able to carry oxygen to your tissues. But it reduces like if you're in regions with malaria, it's very helpful because you don't get the symptoms or you don't get infected because I think you don't get infected. So that's a situation where it seems like a really bad thing. Like your red blood cells are misshapen, and there's this very specific gene that is been isolated for this. So it's like let's get rid of this altogether. But if you're living in a place that still has malaria, then this is you want to have your sickle cells because they actually help you. Even though you're like this seems like a really bad idea, but it is, and this is a very obvious case where there's an advantage to not having a good version, quote unquote, good version of your red blood cells. So, and I think that there's a lot of situations where you get something with one variant and you lose something with that variant, and then in a specific environment, it's like the thrift gene, the quote unquote thrift gene, which is more complicated, but even that idea, 100, 200 years ago, most of the human existence, it was a good thing that you would store more energy if, in a given environment. Now it's like we have so much extra calories that, and then I should guess a little caveat. The thrift gene is people who tend to seem to gain weight with eating, not necessarily a particularly high caloric diet, but for some reason they're very good at extracting calories. And they maybe have a slower metabolism at the same time. So they tend to gain weight in the modern society where calories are very accessible. So in that case, it seems like it's a bad variant. But it was a very good variant when there was no food, which is most of human society, or human existence. So and so that becomes a question with editing, with genetic editing. Is you're editing, what do you lose? What is, like, some stuff where you're gonna, it could cost death within 5 to 10 or 15 years, maybe, whatever the cost benefit there is pretty obvious, but other things that may be less obvious.
Yeah, and my sense as well, and maybe I'm wrong, is this, that maybe the genes or the group of genes that help to raise intelligence are also the group of genes that lead to higher risk of depression, and you kind of just, one goes with the other. There's some good, there's some bad, and you can't just get all upside and no downside.
Yeah, for sure. Sorry, Helen, go ahead.
Well, it's just like, I think that, with what do you call it, with what's the word, exercise, right? Exercise. And so endurance, you tend to, like, you can either be, you tend to have like certain type of fiber types, which are genetically, mostly genetically predisposed, or you get based on genetics. You could be like a really good sprinter, but you're not going to be a good endurance. Like, you're going to lose, you get one and you lose one, right? You can't have it all. You can't, but it's, you're not going to be a really, really good marathon. There's a reason that marathon runners aren't, really good at powerlifting, and it's kind of beyond, just their training. And I think there is an underlying genetic component to it. And that's why elite marathon runners are the way they are, because it allows them to be very good at that, but they lose strength.
Yeah. So look, we've been going on for quite a while now, and I feel like we've covered a lot of ground. And I'm really hoping that we've achieved what we set out at the start. But I'd love to give each of you a final chance to have your say. I'm currently reading Ray Dalio's book, Principles, which I know Phil from PN, the founder of PN, had a hand in making see the light of day. And Ray talks about not knowing what you don't know. So because of my lack of knowledge in this area, they're going to be questions that I didn't know to ask because I didn't know to ask them. So in that idea, like, Krista, is there anything that I didn't ask that you want to add?
No, I wouldn't say that, but it's interesting that you bring up Ray Dalio, because one of his truisms is that we have to face reality as it is. And from the perspective of genetic testing, the reality as it is that we still currently know very little. And the fundamentals, the basics, still apply. So if you're fantasizing that something about your genes makes you spectacularly unique or something like that, it really probably doesn't. And so it's I don't think I would add anything new, but I think I would want to leave people with that reminder that you still have to do all the same stuff that everyone else does, because that's the stuff that we know works. So now, if you're struggling to do that stuff, obviously coaching is a great idea, but you're not going to be so unusual that you have to be doing something fundamentally different. And that is facing the reality of what genes and genetic testing can tell us.
Okay, cool. Helen, is there anything that you wanted me to ask that I didn't?
No, I think I know you did. I think you did a great job. Okay. Especially, for not knowing what you don't know. But no, I think you covered everything and Krista summarized basically where we're at and what I think the take home is. So I don't have anything else to add.
Okay, and Alaina, anything from you?
I'm just going to share my marveling at biology. That this is incredibly complicated, and I think if I want people to take anything away from our book, it's that people who are making claims about what this can tell you, you should be highly skeptical of them, Because the truth is, just like Helen and Krista said, like there's not a lot that we don't know a lot. There's a lot that we don't even know that we don't know. And so you should be tremendously skeptical of anyone who has like jumped on this new bandwagon, just like the paleo bandwagon and the keto bandwagon. Like this genetic bandwagon is another thing. And people come in with like this sort of limited knowledge or understanding of how genetics or evolution work. And then they try to extrapolate, and they will say things that, like, if you don't understand anything about the field, sound plausible. You know, there's a gay gene. Well, that sounds like there are genes and there are gay people, so there must be a gay gene. Well, that's not true, right? But like people who don't have a lot of knowledge will come in and make these sort of grandiose claims. You should be eating a keto diet. You should be eating the Mediterranean diet. And they don't know. Like the people making these claims don't know. And I think you should just be very skeptical of anyone who wants your money based on those claims.
Okay. So look, all of you, thank you so much for coming on the show. I do understand this is not an easy topic to cover, especially doing it at a basic level. And so I think it's been really helpful for people who have come into this knowing absolutely nothing. And then for people who have much more knowledge on this, I still think they would have got something out of it. So just finally, Krista, do you want to point people to where they can get the book or any other links that you want my audience to be aware of?
Yeah, so you can go to precisionnutrition.com and find the book. It's pretty easy. I think the URL is something like genetic testing ebook, but you can just go to precisionnutrition.com and navigate your way to the ebook. And
okay, and I'll put the links in the show notes anyway for it.
Yeah, perfect. Yeah, so if you can find the show notes, you'll find the book. It's completely free. And if you like reading scientific references, there are like 600 for you. So there's hours of enjoyment in this book.
Okay, perfect. Well, thank you again all for coming on the show. I really enjoyed being able to do this.
Thanks for having us.
Yeah, thanks for having us.
Thanks for the opportunity.
Thanks for listening.